Gnanam Paramam Dheyam. Knowledge is supreme.
Hello students, I am sure you are very excited for this upcoming course on applications of interactomics using genomics and proteomics technologies. As you have seen my introduction video that this course would feature several distinguished invited speakers working on the cutting edge areas of genomics and proteomic technologies. Additionally, there will be some laboratory demonstrations which will help your knowledge how to perform these experiments in the laboratory settings. I just thought to interact with you slightly ahead of the course, as the course is going to start from February 25th. It will be a good idea that you should know what will be covered in the course and what could be a good prerequisite to make you better prepared. So, course is going to cover about various technologies which includes protein microarray based platforms like purified protein based arrays, cell free expression based arrays like NAPA, reverse phase arrays, various type of applications used in these technologies, and then we move on to the next generation sequencing technology based platforms. What are the latest advancements happening in these areas from leading companies like Illumina and Thermo Fisher. We also would like to talk to you about label free biosensors, especially surface plasmon resonance, biolayer interferometry and other type of label free biosensor platforms. So, while the course is going to give you a very broad understanding with a combination of various experts from the academia and the industry, it may be good idea that even from now you can start slightly preparing yourself better for this course. In the past I had offered another MOOC course on interactomics where some of the basic technologies were taught. I am going to show you a link, a URL on the screen. Please follow that link and you will have access to some of these videos which I have recorded earlier to talk about some very basic concepts of protein microarray technologies and different type of label free SPR based biosensors. I hope some of those knowledge will really make you well prepared to take this course forward, and when you attend this course while you are watching these lectures, then you will already have the basic familiarity with the previous technologies which we are going to talk. So, it is good idea that in next 2 weeks time what we have, you should start preparing yourself, watch some of the prerequisite videos which will help you to understand these concepts and these applications in much better way. I would also like to re-emphasize that we will be very interactive with you. We would like to interact with you almost every week, once in a week, where we will have a live session from the laboratory directly from my proteomics lab at IIT Bombay, where we would like to show you some interesting experiments. We would also like to take your curious questions and a very good way of engaging each other and trying to interact with you to break the boundaries that it is an online course or it is the virtual course, but rather I want you to feel that we have a physical bonding where we can at least interact and talk and take your queries forward. So, we are very excited to teach you this course in a very short time which is going to be released, and I hope you are also equally excited and you are going to make yourself well prepared to take this course to the best understanding possible, and to do this it is a good idea to start watching some of these videos ahead of time. So, finally, let me again welcome you to this course applications of interactomics using genomics and proteomics technologies, and very soon we will have a lively weekly interactions to talk to you a large number of students who have never met me, but will still have good opportunity for us to interact and take your various interesting questions and try to give you the understanding that how you can use this knowledge for your actual experiments, for your actual research work, or for your understanding for different other courses in future. So, let us get ready to learn some of the very novel, interesting advanced technologies in the field of genomics and proteomics, and let us try to make the best knowledge and the best use of these interactions in the upcoming course. Thank you very much.